Okay, welcome to the first part in the section on BLAST. You'll want to go ahead and download the PowerPoint. Go ahead and start sharing that. Right, so the last part of the course, the previous part that we just finished was on alignment. We started out talking about pairwise alignment and then extended that to methods of multiple alignment. So we did the muscle exercise and then MAFT. But in that section, I glossed over sort of the first step in the sequence about how do you get your sequences, or where do you get them in the first place. Now, obviously, one place you might get them is wet lab experiment, right? Doing the actual sequencing. But more commonly is a situation where you have done some sequencing, so you have your gene of interest, and then you want to go see what it's related to. Or maybe the flip side, which is actually maybe even more common nowadays, is that the organism that you work on just had its genome sequenced, and maybe there's an annotation that has tried to identify the genes in that genome, maybe not, but you know that there are some genes in other organisms, like if you're working in human, well, maybe that's not a good example because it's very well annotated already. But in my case, for example, a new beetle gets sequenced and we have the genome. We know that in the case of an example we're going to use for homework, that there are some genes involved in sex chromosome dosage compensation. And we want to know if this new beetle has those genes. Well, how do you go get those genes, right? So where do we start if we're going to go sort of through that multiple sequence alignment and try to identify conservation, do phylogeny, whatever the case may be downstream? Where do we get that stuff? So the first one on there that I had listed, NCBI, all right, or GenBank is the real repository within NCBI, but that stands for National Center for Biotechnology Information. I give you some links there. This is the all caps primary repository for genetic data. If it's published anywhere, it should be in the NCBI database, and definitely nowadays with the sort of pace of genome sequencing, even if it's not published, it might still be in NCBI. And there's a lot I could unpack there, but for example, especially in these huge genome projects where hundreds and thousands of organisms are having their genome sequenced and assembled, that often becomes available before the actual publication. And there are some sort of formal agreements about using that data in corresponding with the people who generated it, so that nobody gets scooped who has invested time and resources into doing the actual data collection or assembly. So there are lots of tools available. We just don't have time. We could do a whole course on BLAST or some of the tools that are available through NCBI's website all by itself. But there are tutorials available. Great thing about one of the great things about the age we live in, right? There's a YouTube video for everything. So I'm really going to go through today, and in the part 2, maybe part 3, we'll see. Some of the most common tasks. I think we'll have a 3-part series, but I don't know if we'll actually have exercises to do all 3. Today we're gonna just look at, if you have a gene from another organism, trying to identify the homologs. But there are lots of other tools in NCBI. Another major database is Ensembl. This is sort of the European, or or the EU's version of NCBI. There are a lot of tools there. One of the ones that I have used the most is Ensembl. I guess Ensembl falls under the EMBL, EBI. A lot of interesting tools there, especially I have used this one a lot for gene family evolution. So when you have groups of genes that are related to each other and you want to know how copy number changes, there are some nice tools in where they have pre-computed the gene families. A lot of people, so if you just go to the main Ensembl website, you will find in here you can search across some of the main model systems. It's especially good for vertebrates. Okay, so lots of genomes in here, and this has grown tremendously over the last couple of years. Some of these big vertebrate genome sequencing projects. So this one has grown really rapidly, but I just also wanted to point out for those of you who aren't working on a vertebrate, Ensembl also has a separate set of databases for non-vertebrates. So you can go in and look at individual species. This is in the main Ensembl Metazoa, but you can also look at bacteria, protists, fungi, plants, and so the regular Ensembl is the vertebrate one. But if you're interested in or working in some of these other groups, you can go use that specialized Ensembl site. And a lot of the tools overlap, but because of the evolutionary distance between these things, you get a lot more, well, this will become clear as we go through the lecture why you might want to do this, but there are some useful reasons or really important reasons why you might want to limit your searches because of the conservation of genes and sequence within those groups. Okay. So really helpful thing to know is that the main Ensembl site is not the only one. Okay. And the reason I'm highlighting that is that I often get asked, hey, my species is not in Ensembl, what do I do? And people haven't checked to see whether it's in Metazoa or any of these other ones, depending on what organisms you're working on. Oops. Well, in principle, all of the data that's in NCBI is also mirrored to the EU, which is also mirrored to a similar website in Japan, similar database in Japan. And so every night, these things sync with each other. And at least in principle, if you find it in one, it should be in the other. Now, that doesn't mean that all of the tools are available the same. Like I said, I've used the EBI Ensembl website because it has some nice tools for pre-computed gene families, but over time the tools sort of become similar. So tools that develop on one, if they're getting used a lot, they'll move and be deployed on the other servers. But then each of these databases have whole big research programs and teams that are developing tools to help people analyze the data that's in the database. So those teams will develop a new software tool and it will be deployed on their set of servers. And so sometimes you'll find tools that are available on one and not the other. If they turn out to be really useful, then they tend to get migrated over to the other sites. And I give a link if you want to know more about what's going on behind the scenes with these databases. You can read about that collaboration. There are other more specialized databases. If you're in a model organism, for example, there's typically a specialized database that community has developed on its own. All right, there's a the main model system in my lab has been Tribolium. There's a Beetlebase, which has tools and genome sequences for working in the beetle genome. This one is for human, catalog of Mendelian mutations. Disease-associated variants is probably a better way to say it than mutations. Because to be in here, they have to have some kind of association already made with them. So that's where you're going to get the data. So how you're going to get the data in many cases is BLAST. So this is one of, if not the most often cited paper in biology, life sciences, because this is the basic local alignment search tool, was the breakthrough that really allowed us to take a sequence and search for the database. So we talked about pairwise alignment and multiple sequence alignment, and those, especially multiple sequence alignment, even though you can MAFT makes that really fast, it doesn't make it fast enough that you could take your sequence and search the 10 million protein coding sequences that are in NCBI. I actually don't know the real number. It's probably well over that now, but right, that's just not doable to actually do the alignments as a search algorithm. And so that's what BLAST really, the problem that BLAST solved was how do we search for similar sequences. And so this is just a few slides on how BLAST works. So we're going to say this is our query sequence. Right? The thing that the query is the thing that you have that you want to use to search the database. So we want to find matches to this protein coding sequence. So this is just a string of amino acids. Some things that there are default settings when we go to BLAST. But it's useful to know what they are because you might want to modify them based on different kinds of searches. So here our query length is 11, and the word size, which is an important step, is 3. Okay, so basically, the first thing BLAST does is just chop up your query into however large the word size is. So in this case, our word size is 3, so it just slides along. This is exactly the same algorithm that we wrote very early on when we were looking for k-mer matches. Like if we wanted a k-mer count, it's the same deal. So then we're going to generate the neighborhood of words that have a score that has a threshold less than theta. Okay, so totally analogous to what we did when we were counting with mismatches and we had a distance less than d. Okay. The only modification here is that we're moving from the simple scoring matrix that we had where match equals 1, mismatch equals 0. And we're building in those layers that we talked about in pairwise alignment, where we're actually going to score how different two things are. Like in the case of our amino acids, whether you're using PAM or BLOSSOM to say how much should we weight a mismatch between two amino acids. So we're going to use that as our scoring matrix rather than the simple 1 and 0. So you can imagine using that scoring matrix to put into your neighbors function that you wrote in Python to generate all of the things that are some certain threshold distance away. So for 3 amino acids, there are 20 times 20 times 20, because that's the number of amino acids. That's the number of substitutions we could potentially make. So what you would do is, for 3 words, you could expand it to that, except, and I use Blossom 62 here in the example because that's the default for BLAST. And so you just generate all of those potential 3 amino acid sequences, right? Because that's our word size, and then make the cut at whatever your threshold, and in this case, we're going to use a threshold of 10. So everything who has above a score of 10 is what we're going to use as our seeds. So these are the only words that we're going to actually look for. Okay, so again, just so it's clear, right? This is like the analogous thing that we had to do in problem 21 and 23 in the Rosalind problems, where we generated the neighborhood. Okay, in those problems we were using the very simple Hamming distance. In this, all BLAST is doing is using the Blossom 62 scoring matrix to generate that neighborhood. Okay, so that is the word list that we're going to use to search the database. And the thing that makes it really fast, or one of the steps that makes it really fast, right? Generating those neighborhoods, that's fast. That was not one of the slow parts in our solutions for 21 and 23 in Rosalind. Also, we know that if we only just have to look for exact matches, that's also really fast. Those were some of the early Rosalind problems. Right? So you just take those 4 words that we had and search the database for exact matches to those words. Okay. So if you find an exact match, and again, so I'm using words that are small enough here and a query sequence that's small enough to fit it on the screen. Right, so in reality, you might be using like 11 base pair words or something, amino acid words. But here, the idea, right, we find some sequences in the database that have exact matches to our word list. Okay. Then we take each one of those sequences, and starting from the exact match, we start to extend off of the end. And so what I have here is the query, which QEH was our word generated from LEH. So that's where it's matching in the query. And we start to extend KMGS, right? That's the extension this direction. And this blue bar is the sequence in the database. So we're just scoring, right? A match. These values on the bottom come from Blossom 62 matrix. M is a -1, a G, so the MR mismatch is weighted as a -1, GG match is a 6, and the SW is a -3, so then you're summing the score there. And obviously this one in the database, the sequence continues on, but that's the end of our query right there, okay? And so if you look at this graphically, we're going to have a threshold for allowing the mismatch to drop off by 2 or less. So what that looks like here is we went all the way to this SW mismatch. That last one had a score drop of 3, which is greater than our threshold. So we're going to actually not count that in our sort of best hit. So we'll actually best hit maximal segment pair is what we'll call it in a second. So we cut that one off, and really the match ends at the GG in the going to the right. Okay, then we just repeat this same process going to the left, right? So we're getting our individual scores. The graphic here may be a little confusing because we're going HH, and EE is a match, QQ is a match, C, the NP is the first mismatch, so we go down right there, and so on. So this is going to the right, even though this matching is going to the left. The YD is a -3. That's the only one we're going to chop off in this case as well. So really our match goes from that A to that G. And so maximum segment pair is this. Actually, probably, I don't know why I left the Y and the S on there. The maximal segment pair, you would probably, not probably, you would stop at the A and the G. Oh, because I had it on another slide. Right, so that's our maximal segment pair, cutting off the ends. Just realized you probably couldn't see my cursor that whole time. The raw score, right? If you went back to these, and so these scoring is pulled from this row right here, right? And those are the blossom scores for either the matches or mismatches going from A to G. So our raw score here would be 39. Okay. So you're going to do that for all of these things. So all the sequences, so each blue bar is potentially a different sequence, though not necessarily, right? Because LEH and CEH both hit this same sequence, okay? But you're doing that function of extension in both directions and keeping the maximal segment pair for each of the matches, the exact matches that you found in the database. Then once you've got, why is it jumping around? Okay. You've got your raw score, and you've done that across all of those exact matches. And so then you look at, in rank order, what are your maximal segment pairs? So that sequence that had actually 2 seeds on it might give us a much higher score. So you rank all of those things, and then this is what will be reported in your blast hit. So it'll tell you something about the length, something about the score, and that's telling you basically, is this a good match or not? Okay. Well, we're going to talk about how to evaluate that. So some of the data that you're going to get out is one of the ones that you see reported the most often is the E value. So the E value, it's like a probability. It's really the expected value. That's where the E comes from. It's the number of maximal segment pairs that would have similar or higher score than one can expect to see by chance in the particular database that was searched. So the simplest way maybe to understand that is by example, whereas if you have an E value of 1, then you would expect to see at least one match that has a similar length and similar percent identity to that MSP just by chance. So I haven't, the calculation for the E value is there, but for example, if this E value, actually, right? The lower scores are worse matches. And so you would expect things that have a low match to maybe occur at that score just by chance. So if this extended to an e-value of 1, you would say, well, this is probably not a good match, right? Because I would get a match like this or a score like this just by random chance in the database that I searched. Okay, then you get a score up here that has an e-value closer to 0, right? An e-value close to 0 is saying there is 0 chance that you would get a match that good or a score that high purely by random chance. So when we say that those e-values are calculated based on the query length, the size of the database, and the bit score. So I'll talk about bit scores on the next slide, but this is, you can think of it as the score for the moment. The e-value, right, that probability that you're going to see a match like this by chance, hopefully it's somewhat intuitive that it depends on how big is the database, right? Because the bigger the database, the more different kinds of sequence can occur just by random chance. And so you'll get matches that are low quality matches, right? That they're not very similar to each other just by chance because the database is so big. Okay. It's also affected by the length of your query, right? If you have a really long sequence, you're going to get more matches to subsets of that, right? Because remember how it works. You chop it up into little words and then search for matches. And then, right, the more of that you do, the more chance you have of getting a decent quality hit just by random chance. Okay, so that combination of how big is your query and how big is the database, that m times n, the first two variables there, are often called the effective search space, right? The bigger your search space is, the more likely you are to just find matches by chance. So I put this, tried to highlight this as best I could. Got bold, italics, underline. Especially if you're trying to find matches between distantly related things, the smaller you can make your database, the better. Okay. The example I'm going to show today is if I know I'm only looking in, I only want to find homologs in one species, I should just use that species genome as my database, not all of NCBI. And then just one more thing to think about is that how do you compare different searches? Okay. And that is like, if you want to say that one hit is better than another, it's okay to compare the e-value inside an individual search. So if I search gene A in genome for Drosophila melanogaster, all the hits in that search are comparable based on their e-value, okay? Because my query is the same, right? My gene A sequence and the database that I searched is the same. So those e-values are comparable. But as soon as I either change the query, right, the M, or I change the N, right, I search it in a different species, now the e-values are no longer comparable between the first search and the second search. Right? Because these two things are different in terms of, like, if I want to say one hit is better than the top hit in search 1 is better than the top hit in search 2, I can't really make that direct comparison if M and or N have changed. But I can compare the bit scores, right? So that score, how good is the alignment, that is comparable as long as you scale it for the scoring matrix that you used, okay? And so in the output you get from from BLAST, BitScore is the normalized version of that. So I could, between search 1 and search 2, compare, is the score better, right? Is the alignment better? And most, real example, if I have gene A from Drosophila melanogaster and I search Tribolium castaneum in one search, and then I go and search, say, Tribolium confusum, a different beetle, and I want to compare, is the match from Drosophila better in castaneum or better in confusum? I need to use the bit score, not the E value, right? Because the castaneum database is not the same size exactly as the database in confusum. Okay. So again, highlighting the main point there, E values are not directly comparable across searches. They're still telling you within that search, how likely was I to see it by chance?